Placentation in mammals. The placenta is named on the basis of this organ's gross anatomical appearance. A placenta is an organ built up of maternal and fetal tissues jointly. Placenta may be defined as a temporary connection between the maternal and fetal tissues for the purpose of physiological exchange. The placenta functions as a feto-maternal organ with two components: the fetal placenta called chorion frondosome, which develops from the same blastocyst that forms the fetus, and the maternal placenta called decidua basilis, which develops from endometrium. In all viviparous animals, the embryonic development takes place inside the uterus of the mother because the eggs are microlecithal and the amount of stored yolk is not sufficient to cope up with the needs of the developing embryo. Such embryos get attached to the uterine wall to draw essential substances from maternal circulation through the placenta. During the gestation period, it provides nutrition, exchanges gas, removes waste, acts as a source of hemopoietic stem cells, and provides endocrine and immune support for the developing fetus. There are essentially three separate aortic or venous circulatory systems. These are umbilical, systemic, and vitelline. The umbilical system is lost at birth, the vitelline contributes to the portal system, and the systemic is extensively remodeled to form the mature cardiovascular system. The nutrition inside the placenta for the growing embryo is derived from about 100 to 150 maternal uterine spiral arteries located in the basal plate. The human placenta is about 9 cm in diameter. We shall now understand the development of placenta, taking human mammalian embryo as an example. When the embryo enters the uterus, it remains bathed in the uterine fluid containing organic substances produced by the tubular glands of the uterine wall. The early embryo may absorb some of these substances through the epithelial covering till the placenta formation. For its further development, the embryo completely depends on substances supplied to it from the maternal tissue. A mammalian placenta is a composite structure formed from the apposition of extraembryonic membranes such as chorion, allantois or yolk sac with the endometrium of the uterine wall called decidua basilis, which includes both the compact and spongy layers for the purpose of physiological exchange. In between these two parallel plates, a huge blood sinus known as the intervillar space contains an enormous number of chorionic villi. During pregnancy, placentation is formed after the implantation of the embryo into the uterine wall and involves the remodeling of blood vessels in order to supply the needed amount of blood. In humans, placentation takes place 7 to 8 days after fertilization. The chorionic villi from the embryo on the embryonic pole grow, form the chorion frondosome. The villi on the opposite side, that is aembryonic pole, degenerate and form a smooth surface called the chorion lave. The endometrium, called the decidua basilis, from the uterus of mother apposes over the chorion frondosome and forms the decidual plate. The decidual plate is tightly attached to the chorion frondosome and goes on to form the actual placenta. Endometrium on the opposite side to the decidua basilis is the decidua parietalis. This fuses with the chorion lave, thus filling the uterine cavity. In the case of twins, dichorionic placentation refers to the presence of two placentas that is in all dizygotic and some monozygotic twins. Monochorionic placentation occurs when monozygotic twins develop with only one placenta and bears a higher risk of complications during pregnancy. Abnormal placentation can lead to an early termination of pregnancy, for example in preeclampsia. Structure of placenta. In humans, the placenta averages 22 cm in length and 2 to 2.5 cm in thickness, with the center being the thickest and the edges being the thinnest. It typically weighs approximately 500 grams. It has a dark reddish blue or crimson color. It connects to the fetus by an umbilical cord of approximately 55 to 60 cm in length, which contains 2 umbilical arteries and 1 umbilical vein. The umbilical cord inserts into the chorionic plate. Vessels branch out over the surface of the placenta and further divide to form a network covered by a thin layer of cells. This results in the formation of villous tree structures. On the maternal side, these villous tree structures are grouped into lobules called cotyledons. In humans, the placenta usually has a disc shape, but the size varies between different mammalian species. According to the nature of the fetal membrane, there are 3 types of mammalian placenta. Some marsupials like Didelphis or Macropus, allantois remains relatively small and never makes contact with the chorion, whereas the yolk sac with its network of vitelline blood vessels become very large and fuses broadly with the chorion. Further, the chorion never advances beyond a smooth membrane in close apposition with the vascular uterine endometrium. The uterine wall secretes a viscous fluid, the uterine milk. It is absorbed by the villi of yolk sac placenta and through vitelline circulation is carried to the embryo. Some other marsupials like Parameles, Deciduous and all other eutherian mammals, the yolk sac remains rudimentary, but allantoic blood vessels are well developed. The allantois fuses with the chorion to form a composite membrane. This membrane serves as a component part of the so-called Chorio-allantoic placenta. The chorion grows out into root-like vascular processes called the chorionic villi that engage the uterine mucosa in a more or less intimate functional relation persisting throughout pregnancy. When the allantois is insignificant or lacking, the placenta is known as chorionic placenta. This type of placenta is found in human beings. A greatly thickened layer of chorion or trophoblast contains lacunae or sinuses filled with maternal blood. The chorionic villi are finger-like outgrowths that penetrate into depressions in the wall of the uterus, through which all kinds of interchanges takes place between the mother and the fetus. The portion of the trophoblast near to the embryo is called cytotrophoblast or cellular trophoblast and has cuboidal cells. It is also known as the layer of Langhans. The cellular layer gives rise to syncytium called the syncytiotrophoblast, which covers the villi externally and is attached to the deciduous bacillus. The portion of uterine mucosa and epithelium which is reflected over the blastocyst forming a cover or capsule is called decidua capsularis. The remaining part of the uterine wall with which the thin chorion finally comes in contact is known as decidua vera. During the late pregnancy, the cytotrophoblast of the villi becomes progressively more interrupted and hence scarcer, while the syncytium forms the sole covering of the villi. Subsequently, it becomes permeated by a system of cavities called the trophoblastic lacunae. These villi extend, and across the trophoblastic lacunae, the blood-formed capillaries flow into the lacunae, which become available to the vital tissues over very large surface of the trophoblast. According to degree of connection and histological relations between the fetal and maternal tissues, there are 5 types of placenta. The fetal placenta has allantoic blood vessels or vitelline blood vessels. Depending on the degree of connection, there are various types of placenta. Endothelial-chorial placenta. In this type of placentation, the chorionic villi are in contact with the endothelium of maternal blood vessels. The villi push in the wall of the uterus. 6 layers of tissue lie between the fetal and maternal blood streams. They are the endothelial wall of the maternal vessel, the connective tissue around the maternal blood vessel, the uterine epithelium, the epithelium of the chorion, connective tissue of the chorion, and the endothelial wall of the blood vessel in the chorion. It is found in ungulates like horses, donkeys, pigs, and all marsupials including dogs and cats. Syndesmochorial placenta. The fetal and maternal components are fused intimately, destroying the uterine epithelium. The chorion comes in contact with the endometrial connective tissues. It has only 5 barriers and is found in ruminants like camels, goat, cow, etc. Epitheliochorial placentation. Chorionic epithelium comes in direct contact with the endothelial walls of maternal blood vessels. The barriers are now reduced to 4. Such type is characteristic of carnivores like tiger, walrus, cat, etc. Hemochorial placentation. The maternal blood comes in direct contact with the fetal chorion, which does not happen in the other 2 types. It is found in higher order primates, including human, and also in rabbits, guinea pigs, mice, and rats. Hemoendothelial placentation. All the 3 maternal tissues and 2 fetal tissues are completely eroded. The fetal blood vessels dip into blood lacunae of the uterus, leaving only 1 barrier. It is found in rat, guinea pig, and in rabbit. According to the fate of uterine endometrium at partition, there are 3 types of placenta. Non-deciduous placenta or semiplacenta. In case of epitheliochorial placenta, at parturition, the chorionic villi are simply drawn out from the crypts in the wall of the uterus without any damage to the wall, and no bleeding takes place. It is found in horse, zebra, and tapir. In this type, the union of maternal and fetal tissues is so intimate that at parturition not only the fetal component of the placenta is shed, but a variable amount of maternal tissue is torn away, leaving an open wound on the wall of the uterus, and bleeding inevitably occurs. It is seen in most mammals as well as human. The fetal part of the placenta does not shed and remains inside the uterus, where it is broken and absorbed. It is seen in mole and parameles. In preparation for implantation of the blastocyst, the uterine endometrium undergoes decidualization. The increased diameter of blood vessels and straighter flow path both act to increase maternal blood flow to the placenta. The relatively high pressure as the maternal blood fills intervillous space through these spiral arteries bathes the fetal villi in blood, allowing an exchange of gases to take place. Deoxygenated fetal blood passes through umbilical arteries to the placenta. At the junction of umbilical cord and placenta, the umbilical arteries branch radially to form chorionic arteries. Chorionic arteries in turn branch into cotyledon arteries. Vessels eventually branch to form an extensive arteriocapillary venous system, bringing the fetal blood extremely close to the maternal blood, but no intermingling of fetal and maternal blood occurs. The fetal placental circulation is vulnerable and persistent to hypoxia or intermittent hypoxia and reoxygenation, which can lead to generation of excessive free radicals. This may contribute to preeclampsia and other pregnancy complications. It is proposed that melatonin plays a very important role as an antioxidant in the placenta. The placenta, being a temporary organ, is expelled, which begins as a physiological separation from the wall of the uterus during the third stage of labor. The placenta is usually expelled within 15 to 30 minutes of birth. Placental expulsion can be managed actively by giving oxytocin via intramuscular injection followed by cord traction to assist in delivering the placenta. Alternatively, it can be managed expectantly, allowing the placenta to be expelled without medical assistance of extrauterine life, especially in preterm infants. The exchange of fetal and maternal blood allows the transfer of nutrients and oxygen by both active and passive transport from the mother to the fetus. Adverse pregnancy situations such as those involving maternal diabetes or obesity can increase or decrease levels of nutrient transporters in the placenta, resulting in overgrowth or restricted growth of the fetus. Waste products excreted from the fetus such as carbon dioxide, urea, uric acid and creatine are transferred to maternal blood by diffusion across the placenta. IgM antibodies can pass through the human placenta, thereby providing protection to the fetus in uterus. This transfer of antibodies begins as early as the 20th week of gestational age and certainly by the 24th week. This passive immunity lingers for several months after birth, thus providing the newborn with humoral immunity to help the infant survive through the crucial first months of extrauterine life. IgM, however, cannot cross the placenta, which is why some infections acquired during pregnancy can be hazardous for the fetus. The placenta functions as a selective maternal fetal barrier against transmission of microbes, blood proteins, hormones as well as maintain blood pressure differences. However, insufficiency in this function may still cause mother to child transmission of infectious diseases. The placenta stores fat, glycogen and iron for the embryo before its liver is developed. The placenta, besides other functions, also secretes hormones that are essential to fetal life, such as the chorionic gonadotropin hormone, estradiol, progesterone and placental lactogen. These hormones help in maintaining pregnancy, abortion and premature birth. In some mammals like cow and rabbit, it produces relaxin hormone that facilitates birth of young ones. Human placental lactogen is a hormone used in pregnancy to develop fetal metabolism and general growth and development. The placenta and fetus may be regarded as a foreign allograft inside the mother and thus must evade from attack by the mother's immune system. For this purpose, the placenta secretes neurokinin B containing phosphocholine molecules. This is the same mechanism used by parasitic nematodes to avoid detection by the immune system of their host. There is presence of small lymphocytic suppressor cells in the fetus that inhibit maternal cytotoxic T cells by inhibiting the response to interleukins. It can occur following placental infection leading to maternal inflammation of villus stroma. It is often associated with intervillositis. The inflammation can lead to disruption of blood flow and necrotic cell death. The maternal blood-filled space is filled with CD68 positive histiocytes and an increase in fibrin occurs more commonly in the first trimester. Prolonged meconium exposure leads to toxic death of myocytes of placental vessels. A fetal malignancy that leads to an enlarged placenta with tumor cells in the fetal circulation and rarely in the chorionic villi. Protein C or S deficiency, factor V laden sickle cell disease, antiphospholipid antibody can generate an increased fibrin or fibroid deposition in the maternal or intravillar space. This can trap and kill villi. The placenta is an organ that connects the developing fetus to the uterine wall and help in nutrient uptake, waste elimination and gas exchange via the mother's blood supply. It provides thermoregulation to the fetus, fights against internal infection and produces hormones to support pregnancy. The placenta provides oxygen and nutrients to growing babies and removes waste products from the baby's blood. The placenta attaches to the wall of the uterus and the baby's umbilical cord develops from the placenta that connects the mother and the baby. Despite having this critical and fundamental similar function in all mammals, the placenta exhibits striking morphological variations.